The overall purpose of this procedure is to use microscopy as a tool to study the phagocytosis of the pathogenic fungal species Cryptococcus neoformans by its environmental predator amoeba.
This video details a protocol for preparing a co-culture of cryptococcal cells and amoeba, which I will study using confocal laser scanning microscopy and transmission electron microscopy. Information obtained from using these techniques could shed insights into the behavior of cryptococcal cells when internalized during infection.
Streak out the test fungal strains on YPD agar plates. Incubate the plate for 48 hours at 30 degrees Celsius. Scrape off a loopful of cells from the 48-hour-old plate and inoculate them into a 250 milliliter conical flask containing 100 milliliters of YNB broth that is supplemented with 4% glucose. Incubate the flask at 30 degrees Celsius for 24 hours on a rotary shaker. After a 24-hour incubation period, count the fungal cells using a hemocytometer and adjust the cell number to 1 million cells per milliliter with physiological buffer solution.